Hello, my name is DrPaulCottrell, and I'm recording this on 5/7/2026 at approximately 2:30. I'm going to go over in this video the point mutations that were developed to understand how the hantavirus can be moved from a Golgi budding process to a cell surface budding process. All right, we're going to go over this research was published 2019, talking about the actual point mutations that were developed. So this is a gain of function. This is a BSL-2 laboratory gain of function research. So the rodent to human transmission of hantaviruses is associated with severe disease. Currently, no FDA-approved specific antivirals or vaccines are available, and the requirement for high biocontainment BSL-3 laboratories limits hantavirus research. The study hantavirus entry in a BSL-2 laboratory, we set out to generate replication-competent, recombinant vascular stomatitis virus, or RVSV, bearing the GN and the GC. These are the glycoproteins that are on the surface of the hantavirus, entry glycoproteins. As previously reported, RVSVs bearing New World hantavirus GN and GC were readily rescued from complementary DNA. But their counterparts bearing the GN, GC from the old world hantaviruses, Hantavirus, which is from Korea, I believe, and the Dobarva Belgrade virus, or DOBV, were refractory to rescue. However, serial passage, here again, that serial passage pops up. However, serial passage of the rescued RVSV HTNV GN-GC virus markedly increased its infectivity and capacity for cell-to-cell spread. This gain in viral fitness, so gain of function, was associated with the acquisition of 2-point mutations, I532K in the cytoplasmic tail of the GN and S1094L in the membrane proximal stem of GC. Now you have to remember the GN and the GC are a complex that binds together in a tetramer, and part of that spike is outside of the viral membrane, and part of that spike is inside the virus. Follow-up experiments with the RVSVs in single-cell VSV pseudotypes confirm these results. Mechanistic studies revealed that both mutations were determinative and contributed to viral infectivity in a synergistic manner. So when you have both of these mutations, the I532K and the S1094L, they're synergistic. Our findings indicate that the primary mode of action of these mutations is a relocalize HTNV GN GC from the Golgi complex to the cell surface, thereby affording significantly enhanced GN GC incorporation into budding VSV particles. Finally, I532K slash S1094L mutations in DOBV GNC GNGC permitted the rescue of RVSV DOBVs, GNGC, demonstrating that incorporation of the cognate mutations into other hantaviral GNGC proteins could afford the generation of RVSVs that are otherwise challenging to rescue. The robust replication competent RVSVs bearing HTNVs and the DOBV GNGC reported here and may also have utility as vaccines. All right, the main point that I'm making here is that one, they have found out that the hantavirus will bud closer to the cell surface, will move its glycoprotein to the cell surface and chaperone the long, small, and medium segments of the genome to allow for it to bud at the cell surface instead of budding off of the Golgi apparatus. Now, the old world viruses typically bud off of the Golgi apparatus, while the new world hantaviruses have a tendency, especially the Andes, to bud off of the cell surface instead of the Golgi apparatus. So old world, primarily Golgi apparatus. New world, some of the hantaviruses will bud off the Golgi apparatus and then do a exocytosis. But with certain types of mutations, the budding will be at the cell surface and not at the Golgi apparatus. That's the key. That's the most important thing out of this. And that two mutations that are important to allow for that to happen are the I532K and the S1094L mutations. Now, when you have these mutations, also in some of these older viruses, like DOBV, that are not easy to rescue, then you improve the ability to rescue them. Because one, they're able to exocytose out of I should say that they're budding off of the cell more often or easier with this mutation. So this method to study these viruses can be used for this platform. All right. So it is known that the Andes hantavirus, has the ability to bud off of the cell surface more often than actually needing the Golgi apparatus. So there's been gain of function that's been taking place at the laboratory that, one, allows us to understand what's happening in nature, but also the ability to take these viruses and to enhance them, to make them more viral. So it's important to understand that there are two mutations that they have developed in 2019. That's the I532K and the S1094L mutations that actually changes the relocalization of the GNGC and moves that to the cell surface. And the chaperoning of the genome, the three segments, the long, the medium, and the short segment of the hantavirus genome, then assembles at the cell surface instead of in the Golgi apparatus and buds off of the cell. And this is the main reason that Andes is more of a human-to-human transmission, but it's probably not the only reason. But the point here is that where it's assembled in the cell surface gives it different capability in terms of infectivity in humans. So if there's more of a viral load, or it's easier for assembly to spread to other cells, it becomes more of a problem. If it's spreading faster in your kidneys or in your lungs, then obviously you're going to have these problems that are pulmonary and cardiac and even renal problems. This is that renal syndrome, this HFRS. This is that hantavirus pulmonary syndrome in the Americas and hemorrhagic fever with renal syndrome in Eurasia. So it's a little bit more pulmonary in the New World. It's more renal in Europe and in Asia. And in the New World, it is more of a budding off of the cell surface where the assembly is taking place. And in the Old World, hantaviruses, they are primarily assembled through the Golgi apparatus. So that's the key takeaway from this research. Please help support the work that I do by subscribing to all my channels. I have lots of different modalities that you can watch my work on. So take a look at all the links are in the description. And go to my website, the-studio-reykjavik.com. Help support the work that I do by donating. You can donate through Stripe, PayPal, or Buy Me a Coffee. You can also be a paid member to my Patreon channel. I do all these videos for free. So it's the help of individuals that want to help grow the channel and help support the work that I do that allows me to do this. So thank you for listening. And for the ones that have been contributing and helping me with producing this content, I greatly appreciate it. And also take a look on my website, the great products that I offer. Thank you for listening and have a nice day.